I'd like to turn our attention now to a little bit towards the future and to talk about particularly what we heard at this ASH 2014 meeting about promising new therapies. And I'm going to start with you, Dr. Kumar, because you presented results on the use of a novel proteasome inhibitor. So you want to tell us a little bit about that and where that's heading?
Sure. I think proteasome inhibitor as a class is going to be an integral part of myeloma therapy for years to come, decades to come. The biggest problem that we have with the proteasome inhibitor right now is patients have to make a trip to the clinic, whether it's Velcade or whether you're talking about carfilzomib and infusion, subcutaneous injections. The ability to give something orally would certainly change the paradigm in the disease now that we can develop very effective all oral combinations, especially since the proteasome inhibitor immunomodulatory combinations are probably one of the most effective regimens as we talked about. So Ixazomib is something that's similar to bortezomib but also has some distinct characteristics to it in terms of its properties. And this is the first oral proteasome inhibitor to be in the clinic in trials. The data that this has already been studied in the relapse setting as a single agent and has been shown to be active even in patients who have previously seen bortezomib. And we know what dose to give, how often to give. It's given once a week for 3 weeks and then a week off. The particular study looked at trying to combine the Ixazomib with lenalidomide and dexamethasone in newly diagnosed patients. We saw very good efficacy. 90% of the patients had response. 60% of the patients had a very good partial response. It was well tolerated. The main side effects we saw were skin rash and gastrointestinal side effects. More than the efficacy in the induction phase, we also found that we could give this drug as a maintenance drug, at least up to 2 years is what we have seen in the study, with some deepening of the response over time without necessarily any added toxicity. So I think it represents oral proteasome inhibitors as a group certainly represents the way to go in this disease. So we have other oral proteasome inhibitors being developed in the clinical trials as well.
And what's the path for Ixazomib? Is it in late stage testing or what's happening with the drug?
So there are multiple phase 3 trials ongoing. Clearly it's being looked at in combination with lenalidomide and dexamethasone versus lenalidomide and dexamethasone in the relapsed patient population. That trial is already completed accrual. There's another phase 3 trial that's looking at the same combination comparison in the newly diagnosed setting that's currently accruing. But I think there's one of the unique properties to this oral proteasome inhibitors would be the maintenance space. And there are clinical trials ongoing in that phase as well, looking at both post-transplant and non-transplant arena.
So it's been pretty broadly investigated. What do others think on the panel about Ixazomib impressions, Dr. Jagannath?
I think I had the opportunity to participate in the clinical trial, and it is fairly well tolerated. And one of the trial I participated was once a week oral regimen. I mean, that's very, very convenient. The noise, since it is a bortezomib derivative, the peripheral neuropathy noise was very low. Granted, in the study I participated was on a once a week, and even bortezomib once a week has a lower threshold for neuropathy. But I was quite impressed. The activity is good. So now you're able to use a proteasome inhibitor, and patients don't have to travel back and forth to the center.
So it really sounds like we might have something a lot more convenient for our patients, similar mechanism of action in late stage testing already. Let me turn now a little bit to, I think, something we're all excited about, which is monoclonal antibodies. And these are myeloma, unlike most other malignancies, doesn't have a monoclonal antibody yet. And the first one we're going to talk about is called Elotuzumab. And maybe, Dr. Lonial, you've been heavily involved in Elotuzumab. Can you tell us a little bit about what the antibody is and a little bit of a sense of what we're seeing in the clinic?
Elotuzumab is a monoclonal antibody whose target is something called SLAMF7. And SLAMF7 is a receptor present on the surface of almost all plasma cells, as well as slightly represented on NK cells as well. And what we learned about Elotuzumab as a single agent in the refractory myeloma setting was that it really induced stabilization of disease for the most part, not a lot of overwhelming single agent activity. But when combined with lenalidomide and low-dose dexamethasone, the response rate jumped to 80+%. The durability and the overall survival for those patients in a phase 1 study was really quite striking. And so at the ASH 2014 meeting, Paul Richardson reported on a long-term update of the low dose of Elo, 10 milligrams per kilogram versus 20 milligrams per kilogram, both with lenalidomide and dexamethasone. And what struck me from this long-term update is that the median progression-free survival for the 10 mg per kg of Elo plus len low-dose dex was 33 months. Which again, when you put that in the context of what we're seeing, certainly from Aspire, it's certainly in the same ballpark. Very, very long progression-free survival, and it really brings in a new mechanism. I think you know, we've talked about proteasome inhibitors and IMiDs. Having an antibody would really be very, very nice for the patient.
What about toxicity? You didn't talk to that. Is there any?
Well, so the most common toxicity you see, I think, with all the antibodies we're going to talk about is infusion-related reactions. And the frequency and extent of those are really no different than what we see with antibodies like rituximab or other antibodies in oncology. And that's typically a first or second dose effect. Once you get beyond those, the risk of those goes down markedly.
And just tell us what's happening with this drug. Where is it at in clinical testing?
So there are 2 large randomized phase 3 trials that are complete to accrual. One of them is a relapse trial of Elo-Len-Dex versus Len-Dex, and we hope to have data on that trial within the next 6 to 12 months. The other is a newly diagnosed transplant-ineligible trial, Elo-Len-Dex versus Len-Dex. That one completed accrual in the last year. And then there are other trials that are developing. So Paul Richardson is doing an RVD plus Elo for newly diagnosed myeloma. At the ASH meeting, Saad Usmani presented some data on a SWOG trial of high-risk myeloma with RVD plus Elotuzumab as well. So it's being looked at in a number of different areas. There's smoldering data as well.
So 2015 is going to be a big year for Elotuzumab, it sounds like.
Yeah.
Dr. Berenson, how do you see, if Elotuzumab is as successful as the phase 2 trials would suggest it will be, how do you think this will be used in clinical practice if it becomes an approved drug?
Well, we are currently studying it with the combination of Rev-Dex, trying to give it more conveniently. One of its major drawbacks is the long infusions. We're now currently doing a trial giving it much more quickly, and we'll hope to report that on ASCO 2015. I believe if we can get it more conveniently over a briefer period of time, and if we can show it's safe and well tolerated, which it looks to be, you could argue it may supplant some of these other drugs which have a lot of side effects. This drug doesn't seem to have any outside the infusion reaction.
A drug for relapse or for newly diagnosed patients or for maintenance or for all of the above?
I think the sky's the limit, assuming that they really, the problem that they have right now in my view is the trials they've done have been as a single agent. We heard from the work that Sagar did, there's no activity. And they really are stepping up the plate now with an RD versus ERD trial. So we'll see. And the other question, of course, is can they do pickup sticks on Revlimid failures? I think they need to see if this drug can work in Revlimid refractories. That'll be very interesting.
So Dr. Jagannath, you've told me you're quite excited about the use of Elotuzumab. What makes you feel that way?
Well, the reason I'm really excited is that the patients who went on the trial, which Sagar just now alluded to in phase 1 trial, many of these patients are still ongoing. It's almost now 4th year they are still on the drug. To me, that is really remarkable, the durability of the response. And a couple of those patients actually had adverse cytogenetics. So I wonder whether, using antibodies, we may be able to somehow overcome some of the adverse cytogenetic risk factors which are not quite amenable to the chemotherapeutic operation. So that's why I'm quite excited about it. And in terms of where it'll be used, you heard that there are clinical trials being done in frontline therapy. I think that would be an ideal setting. But right now, the relapse data will come out. So probably in first relapse setting, this antibody will play a key role.
I was at a press conference at this meeting, and Tom Martin from the University of California, San Francisco, described monoclonal antibodies as blockbuster drugs. Do you think Elotuzumab could be a blockbuster drug in myeloma?
Elotuzumab is, if it is the first drug to come into the market, as the first antibody, is definitely would be a change in paradigm. And we hope there are other monoclonal antibody which you would want us to discuss, so we can come at the end of it to perhaps answer your question again.
Now, you mentioned